Hi, good day. I'm Ma'am Strawberry Versosa, and for this session, we are going to discuss topic from our Pivot 4A Learner's Packet or LEAP. And this is DNA replication and protein synthesis. Are you ready? Before we go on our lesson proper, let's have here our most essential learning competencies or MELCs. We have here explained how protein is made using information from DNA and explain how mutations may cause changes in the structure and function of a protein. First, let's differentiate DNA and RNA structure. DNA stands for deoxyribonucleic acid. So our DNA is a nucleic acid that stores and transmits the genetic information from one generation of an organism to the next. And also, DNA is a double helix or double-stranded molecule composed of a complementary strands of deoxyribonucleotides units. Our building blocks of DNA is what we call nucleotides. And each nucleotide is composed of one phosphate group, sugar deoxyribose, and one of the four bases adenine, thymine, cytosine, and guanine. Across the strand of the helix, so always remember that our adenine always pairs with the thymine, and thymine pairs with adenine, and our cytosine pairs with the guanine, and guanine pairs with cytosine. So again, always remember ATCG. Also, our DNA is located in our nucleus or in the nucleus. So we have here our RNA. So our RNA is our ribonucleic acid. Like the DNA, RNA is also a nucleic acid. Unlike our DNA, RNA is single-stranded molecule. In our DNA again, our DNA use deoxyribose sugar while our RNA use sugar ribose. Then, it contains 4 nitrogenous bases, but instead of thymine, RNA contains a similar base called uracil. The uracil pairs with adenine. So instead of ATCG in our RNA, we are going to use AUCG. Adenine pairs with uracil, then ganun din sa ating cytosine and guanine. Again, uracil pairs with adenine. There are major types of RNA. It includes mRNA or the messenger RNA, rRNA or the ribosomal RNA, and the transfer RNA or the tRNA. Then, kung ang ating DNA is located in the nucleus, ang ating RNA is located in the cytoplasm. So, DNA carries the information to make proteins. So, how does it work? So, paano nga ba? Okay? So, we have here, so ito napaka-importante. The central dogma of molecular biology or the central dogma of life. Francis Crick and James Watson are credited with this discovery. So, sabi nga nila, detailed residue by residue transferred of sequential information. So, the DNA copies itself by the process of replication, and the DNA copied to RNA in the process of transcription, and finally, you can use the information in the RNA to synthesize a protein in a process called translation. So, to make it simple, so we have here DNA makes RNA and RNA makes proteins. So, why is DNA replication important? So, The process of making copies of DNA is what we call the DNA replication or replication. The DNA copied during the interphase prior to mitosis and meiosis. It is important that the new copies are exactly like the original molecules. The structure of DNA provides a mechanism for making accurate copies of the molecules. When DNA replicates, two identical copies of DNA molecules are produced, which is exactly the same as the original. This also ensures that every new generations of individuals has the same genetic information as his or her parents. So kaya usong-uso ngayon sa mga teleserye yung mga DNA testing. What do you think is the role of the RNA in making proteins in the cell? Can you imagine a car being assembled in the car factory? By way of analogy, different cars are being built in many simple steps. Engineers tell workers how to make cars, and the worker follow directions to build the cars. Suppliers bring parts to the factory so they can be installed in the car. Protein production is similar to car production. It is the role of the DNA to provide worker with instruction for making the proteins, and the workers build the proteins. Other workers, known as the amino acid, bring parts to the factory. RNA molecules, or the workers for protein synthesis, get the instructions from the DNA on how the protein should be assembled. Moreover, do you know that there are 3 types of RNA that help build the proteins? You can consider this RNA molecule to be worker in the production of protein. So we have here the RNA. So we have the mRNA or the messenger RNA, rRNA or the ribosomal RNA, then the tRNA or the transfer RNA. In our mRNA, it brings the information from the DNA in the nucleus to the protein manufacturing area, which is the cytoplasm. Again, the protein manufacturing area is the cytoplasm. Okay, the mRNA becomes the template of information to make proteins. Then, the rRNA or the ribosomal RNA hold tightly into the mRNA using its information, assemble the amino acid in correct order. Lastly, we have the tRNA or the transfer RNA. The tRNA supplies amino acids to the ribosome to be assembled as protein. So, did you find this car-making analogy helpful? But how does the information in DNA, which is found in the nucleus, move to the ribosome in the cytoplasm? So, let's talk about the transcription. Okay? RNA molecules are produced by copying part of a nucleotide sequence of DNA in a complementary sequence in RNA. So this is what we call the process of transcription. So what are the processes of transcription? We have ribonucleic acid polymerase enzyme bind and opens the DNA molecules that will be transcribed. As the DNA molecule opens, the RNA polymerase slides along the DNA strand and links free RNA nucleotides that pair with nitrogenous bases of the complementary DNA strand. Hence, if the sequence of the bases on the DNA strand were cytosine, cytosine, guanine, thymine, adenine, cytosine, adenine, thymine, or CCG, TTA, CAT, the sequence of bases on the RNA strand will be, so ano kaya? So again, remember, AUCG in our RNA. AUCG. So we have guanine, cytosine, adenine, uracil, guanine, uracil, adenine, or GGC, AAU, GUA. When the process of base pairing is completed, the RNA molecule breaks away as the DNA strands rejoin. The RNA leaves the nucleus and goes to the cytoplasm. So don't confuse, okay, in replication with transcription. So in DNA replication, DNA and DNA. In our transcription, we have DNA and mRNA. So in our replication, we have the original strand of the DNA. We have ACGT ng kanyang complement ay TGCA. Wherein sa ating transcription, we have the DNA and mRNA. So, ACGT. So, ang ka-complement niyang mRNA is UGCA. So, let's now move on in translation. The process of converting the information in mRNA in a sequence of amino acids that makes a protein. It is a decoding of messenger RNA in a protein. The transfer RNA or tRNA carries amino acids from the cytoplasm to ribosome. Okay, so we have a series of 3 adjacent bases in a messenger RNA molecule codes for a specific amino acid. It is called the codon. Each transfer RNA has 3 nucleotides that are complementary to codon in mRNA. So it is what we call the anticodon. The processes of translation. As translation begins, mRNA binds to a ribosome. The transfer RNA molecules, each carrying a specific amino acid, approach the ribosomes. The transfer RNA anticodon pairs with the first mRNA or the start codon, adenine, uracil, guanine or AUG, to form the initiation complex. The two molecules temporarily join together. Usually, the first codon on messenger RNA is AUG, which codes for the amino acid methionine. AUG signals the start of protein synthesis. Then, the ribosome slides along the messenger RNA to the next codon. A new tRNA molecule carrying an amino acid pairs with the second mRNA codon. When the first and the second amino acid are in place, an enzyme joins them by forming a peptide bond between them. As the process continues, a chain of amino acids is formed until the ribosome reaches a stop codon. For example of the stop codon is UAA, UAG, UGA on the mRNA strand. The polypeptide chain is released. Protein synthesis is completed. So let's have here an example. Okay, so we have here mRNA, the tRNA, and the amino acid. Okay, so again, sa mRNA natin, makukuha ang ating codon. So, ang ating codon, we have here the first set of codon, AUG, na may ka-complement siyang tRNA, which is the anticodon, UAC. na yung ating tRNA ang magke-carry ng amino acid na ma-form o makukuha natin sa ating codon na AUG. Again, ang tRNA natin ang magke-carry ng amino acid na methionine na na-form from the codon AUG. So, paano nakuha ang ating methionine o methionine? So, we have here the genetic code table. Okay? So, we have here first base or the first letter, second base or the second letter, and the third base or the third letter. First, we have the A, first base. So, dito tayo kukuha ng ating first letter. Next letter is U. So, dito sa second base. So, dito siya maglalay. Then, lastly, in our first codon is G. Okay? So, dito nag-intersect yung tatlo. So, we have the methionine. Okay? So, next one is the UUC. So, ating mRNA UUC. So, we have the codon UUC na ang kanyang magiging anti-codon is AAG. Again, tRNA AAG, siya ang magke-carry ng amino acid na ma-form sa UUC na ito yung phenylalanine. Paano makuha ang phenylalanine? Our first letter is U. U. Our second letter is U. So, dito din. Then, our third letter is C. So, dito sila naglay. So, we have the phenylalanine o alanine. Okay? Lastly, or our last example, so we have AAA in our codon na ang kanyang tRNA o anti-codon is UUU na ang kanyang mapo-form is na amino acid is lysine. So, paano yon? So, we have the codon AAA. A. Then, second letter or second base is A. Then, last or the third is A. So, saan naglay? So, sa lysine. Okay? Sana nakuha yon. Okay, why it is important that a stop codon be part of a protein synthesis? So, kung napansin nyo dyan, meron tayo dyang mga stop codon. Stop yung ating UAA, UAG, UGA. So, bakit kaya? Why it is important that a stop codon be a part of a protein synthesis? Okay, when a stop codon is reached, translation ends and amino acid strand is released from the ribosomes. Okay? So, let's have another exercise. Find the amino acid sequence coded by the following mRNA strand. Okay, wag kayong umiyak o wag kayong mahirapan, wag tayong mahirapan kasi hindi naman natin kailangan sauluhin o memorize yung ating genetic code table. Okay? May guide naman tayo dyan sa ating genetic code table. Okay? So, let's have here the first example. So, we have the mRNA strand. We have CAC o the cytosine, adenine, cytosine, followed by the second codon, set of codon na cytosine, cytosine, adenine, third codon, uracil, guanine, guanine, and uracil, guanine, adenine. Then, our another set of mRNA strand, we have the adenine, uracil, guanine, adenine, adenine, cytosine, guanine, adenine, cytosine, uracil, adenine, adenine. So, what will be the amino acid na mapo-form dito sa ating mRNA strand na to? Sa CAC, we have the histidine, CCA, proline, UGG, tryptophan, then UGA, stop or stop codon. Then, we have here the AUG, AAC, GAC, UAA na methionine, asparagine, aspartic acid, and stop codon. Okay? So again, given itong ating table, genetic code table. Okay? Basta alam lang natin kung paano makuha or yung tamang pagkuha o pagtingin ng ating first base or first letter, second base, second letter, and the third base, our third letter. Okay, so let's have transcribe and translate this DNA strand without looking at your notes. Tingnan natin. We have here the DNA strand TAC, AGT, ACC, AT, AATC, or we have the thymine, adenine, cytosine, adenine, guanine, thymine, adenine, cytosine, cytosine, adenine, thymine, adenine, adenine, thymine, cytosine. So what will be the complementary mRNA, tRNA, and amino acid ng ating DNA strand na ito? So in our mRNA, we have the AUG, UCA, UGG, UA, UAU, UAG. Okay? Or the adenine, uracil, guanine, uracil, cytosine, adenine, uracil, guanine, guanine, uracil, adenine, uracil, uracil, adenine, guanine. Then, ang kanyang tRNA ay UAC, AGU, ACC, AUA, AUC. So, nasusundan? Or we have uracil, adenine, cytosine, adenine, guanine, uracil, adenine, cytosine, cytosine, adenine, uracil, adenine, adenine, uracil, cytosine. Then, ano ang mapo-form na ating amino acid? So, again, ang amino acid natin ay dito natin kukuhanin sa mRNA, hindi dito sa ating tRNA. Again, ang tRNA natin, yan ang magke-carry ng amino acid na mapo-form sa ating mRNA. So, AUG, dito natin kukuhanin yung codon natin. AUG, UCA, UGG, UAU, UAG. So, we have the methionine o methionine, serine, tryptophan, tyrosine, then stop. Okay? May tinatawag tayong mutation. So, ano ba ang mutation? It is change in the base sequence of the DNA. It may affect only one gene or they may affect the whole chromosomes. Mutation in an egg or sperm affect future generation by transmitting these changes to the offsprings. While mutation in the body cell affect only the individual or not passed on the offspring. There are two types of mutation. We have the gene mutation, which is permanent change in DNA sequence that makes up a gene, and a chromosomal mutation, which occur at the chromosomal level resulting in a gene deletion, duplication, rearrangement that may occur during the cell cycle and meiosis. It may cause by parts of chromosomes breaking off or rejoining incorrectly. Most mutations are harmful. Some mutations in the body cells are known to cause cancer, while mutation in sex cells can cause birth defect. A severe mutation may lead to cell death and may have no effect to the body. Some mutations may be useful for the species. For example, a mutation in the blood proteins prevent virus and parasite to thrive in the host organism. Consider what might happen if an incorrect amino acid was inserted in a growing protein chain during the process of translation. Do you think this will affect the structure of the entire molecule? This can possibly happen in point mutation where a change in a single base pair occur. Read the two sentence. What happens when a single letter in a first sentence is changed? The dog beat the cat. And the dog beat the car. Did you see that the changing a single letter also changes the meaning of the sentence? A change in nitrogenous base in a protein may yield a different amino acid and a corresponding change in a protein structure and function. So in our week 4, we have different tasks. So we have learning task number 1. We have DNA versus RNA. So fill in the comparison table below. So we have basis of comparison. We have the DNA and RNA. So number 1, number of strands. So ilan ang strands ng DNA and RNA. Number 2, location in the cell. Saan kaya nalolocate o nasaan ang ating DNA and RNA? Number 3, type of sugar ng DNA and RNA. Number 4, nitrogenous base pair of DNA and RNA. Then, in our learning task number 2, true or false. Write the word true if the statement is correct, and if false, change the underlined word or words to make the statement correct. So, we have number 1, a molecular group consisting a sugar molecule, a phosphate group, and a nitrogenous base is a nucleic acid. Number 2, in a DNA molecule, a base pair would be composed of adenine and guanine. Number 3, cytosine and adenine are purines. Number 4, during the replication of DNA, bonds are broken between the phosphate groups. Number 5, RNA contains the nitrogenous base uracil. Number 6, there are 20 different amino acids in a cell. 7, coded message carried by messenger RNA is translated into polypeptides at organelles called chloroplasts. 8, the decoding of messenger RNA message into a polypeptide chain is known as translation. 9, a severe mutation may lead to cell death and may have no effect on the body. Number 10, most mutations are harmful. Then, we have here learning task number 3. Trace the code. Identify the amino acid coded by the mRNA codon using the genetic code table. So, we have here order of bases in DNA, order of base in mRNA or our codon, order of bases in our transfer RNA, then or our anti-codon, then amino acid coded into proteins. So, i-fill nyo yung mga missing. Okay, in our learning task number 4, essay, explain how protein is made using the information from DNA. So again, explain how protein is made using the information from DNA. Learning task number 5, multiple choice. Choose the letter of the correct answer that best completes each statement and write it on the space provided before each number. Number 1, RNA that carries the genetic code to the ribosome. A, mRNA, B, tRNA, C, rRNA, D, RNA polymerase. Number 2, DNA is a blank. A. Carbohydrate, B. Lipids, C. Nucleic acid, D. Sterol. 3. A nucleotide of DNA would contain A. Uracil, deoxyribose, and phosphate, B. Phosphate, deoxyribose, and thymine, C. Nitrogenous base, phosphate, and glucose, D. Adenine, ribose, and phosphate. Number 4. Watson and Crick described the DNA molecule as A. Branching chain, B. Straight chain, C. Single strand, D. Double helix. Number 5. Between which type of compound in a double-stranded DNA molecule must the bond break before replication take place? A. Phosphate base, B. Sugar phosphate, C. Adenine thymine, D. Sugar base. 6. To which organelle does messenger RNA attach? A. Chloroplast, B. Ribosomes, C. Mitochondria, D. Lysosomes. 7. The coded information in a DNA molecule directly determines the formation of blank. A. Polysaccharides, B. Lipids, C. Monosaccharides, D. Polypeptides. 8. If the code for an amino acid is ATG on the DNA molecule, this code on the transfer RNA molecule may be written as A. ATG, B. CTG, C. AUG, D. CTA. 9. It is the process of making mRNA or messenger RNA strand from a DNA template. A. Replication, B. Transcription, C. Translation, D. Protein synthesis. 10. Transcription takes place in blank. A. Cytoplasm, B. Chloroplast, C. Nucleus, D. Mitochondria. So in our reflection, so on your notebook or on your pad paper, so write, I understand that, I realize that, and I need to learn more about. Okay, so in our references, we have the Science Learner Materials pages 267 to 283. Then Biology Miller Levin New Edition pages 137 to 155. Then special thanks to the author of the Learner's Packet Leaf week number 4, Elpidio L. Vidalio Jr. and Nicanor O. Reyes II. Okay? Kung wala nang tanong, so sana ay may natutunan tayo. Okay? So see you by next session natin sa ating next topic. So sana ay tuloy-tuloy ang ating learning even though tayo ay virtual na, tayo ay nag-lesson. So stay safe tayong lahat. Okay? God bless.